In this video, we are going to look at how proteins move and how that correlates with function. So looking forward to it and hope you are too. So this is broadly defined as protein dynamics, the dynamism, the dynamics of proteins. So before we get into that, I should want to ask a different question, which is just think for a moment about how we draw proteins and maybe how we've drawn it in these videos, how you've seen it in other classes, but sort of what sorts of mental models are we creating when we're drawing proteins? So maybe actually pause and think about that for a minute. So I'm giving you a chance to pause. Now you've unpaused, great. And here's some of the things, right? So we can draw them in the cartoon form. Come on, pen. There, cartoon form over here, right? This very schematicized representation. Maybe we're interested in things like backbone hydrogen bonds, so we use this representation here. Or we're interested in every single atom in bond, we use this here, or some kind of space-filling model. But there are actually other ways people draw them, right? They might draw them as a circle. If you're a cell biologist just describing some kind of network in a cell, maybe all you need is to say there is a protein here. It doesn't really matter what its structural features are. Or if you're at the sort of more physicsy side of things, maybe you write out some protein as an equation, right? You say, oh, there's a velocity of some reaction and some enzyme concentration and some theta. And so you're actually using a mathematical model to describe how the protein works. But what I'd like to do for the moment, sort of motivating this protein dynamics thing, is to ask the question, well, what does a protein look like to itself? Which is maybe a little bit silly, but I think it's actually a really helpful approach. Like if we could look at a protein on a scale that's relevant to the protein, what would we see? And so a good way to do this is through one of these sort of scale zooming exercises. So let's start with something that we've all seen before. Hopefully we have all at some point in our lives seen a human fingernail. And what I've done is drawn a very tiny box. I'm going to zoom in so we can see it. Very tiny box that we're going to zoom in on. So let's grab that tiny box, zoom in. And what we can see here is we would, at that scale, if we could zoom in and really have that feel our field, we would see individual cells. We'd see keratinocytes, which are the cells that actually generate things like keratin in nails. Cool. But we're still not down to the protein level. So now we're going to draw another little tiny box right there, if it's not obvious. We're going to zoom in on that. And now we're about the scale of these David Goodsell paintings I keep showing. So now we're at a David Goodsell scale, and he's painting proteins. So we've got a protein right there that we can look at. Okay, so now what happens if we jump down the next scale? We actually jump and look at the scale of the protein. Think about for a second what you expect to see. Okay, you ready? You have your mental expectation. This is what I think you'd see. Is that what you expected? What even is going on here, right? And if you've watched my previous videos, you'll probably have an inkling of this. What you're seeing here is a whole bunch of water. It turns out that the level of a protein, right? A protein is about the same size as water. So if we drew all the water molecules, you just wouldn't be able to see anything at all. What I just did is I erased the waters, I left them in. I also left in some ions. So those ions in purple and green will let us sort of see where the water molecules are going without completely obscuring our view. But now we can see, oh, there's the protein, all of its atoms arranged in a certain way. And now we can see it bouncing around. And hopefully what comes out when you're looking at this is this doesn't really look like any of the pictures that we've drawn, right? It's bouncing around, it's spinning, it's squishing in on itself. There's all kinds of random motion happening. And that's because proteins are small, which means they're getting bounced around by water. In an earlier video, I imagined it as, imagine yourself surrounded by softballs being shaken in a martini mixer, right? That's how aggressively bouncy the experience of a water molecule is, or sorry, of a protein in water molecules is. And so you have this bouncing around randomness that's going on. They're also moving very fast. So most of these motions are happening on the order of 10 to the 15th to 10 to the minus third seconds. So these are all very quick things relative to our experience while they're being random. So what I'd like to do in the next sort of chunk of this video is unpack some of those dynamics. What sorts of things are happening on what time scale, and how does that actually relate to protein function? So at the very shortest time scales, what we have are things like bond vibrations. So bonds and stretching, or in this case, I've drawn sort of a bending mode for bonds, but there's all vibrations that are happening sort of below the 10 to the minus 12th second time scale. The next time scale up, say around the nanosecond time scale, what we're seeing are things like side chains rotating. In fact, those movies that I was showing of everything bouncing around, that's sampling about the nanosecond time scale. So if we zoomed in on side chains, we would have seen they're rotating all over the place. And then the next level of motion up are these kind of loop motions. So this is, ranging from, I don't know, tens of nanoseconds up to milliseconds. And I'm showing this in kind of a smoothed out form for a protein. And you can see it sort of Pac-Manning along. And so hopefully what you can appreciate is there's all kinds of motion across a bunch of scales. And also that it's actually a pretty wide range, right? This is 10 to the minus 15th to the 10 to the minus 3rd. So there's actually 12 orders of magnitude of scale of motions happening here. So there's, motion, depending on what we look at, we can see all kinds of different sorts of processes happening. Okay, one way that people describe this, and I think a really helpful way to look at this, is describing this as a protein energy landscape. So hopefully you've seen diagrams before where we have an energy axis, and then I'm going to draw the one you might have actually seen. You might have seen something that looks like this, right? Or maybe an enzyme that's showing, or a reaction showing that you have some reactant over here and some product over here, and it has to hop over some energy barrier. In this case, you've got two possibilities. You've got 1 and 2, the reactant and product, and then you're on some energy landscape. Well, in this case, what we have is the same sort of idea. We've got an energy landscape, but now instead of 1 and 2 states, you've got a whole bunch of different conformations. And so a protein that's sitting around in the solution is actually flopping back and forth between these, and it's spending more time down at the bottom at the lower energy conformations, the lower energy structures, and then it occasionally hops up here and spends time at the higher energy structures. And basically every protein that you look at is going to be flopping around amongst multiple structures as governed by its energy landscape. In addition to having a cool energy landscape, which I just think is a fun idea to think about, the motions there we're talking about are actually often quite functionally important. And so this is the loop motion picture that I showed. This is actually an enzyme we've already seen many times already in this set of videos. It's called staph nuclease. It is an enzyme that cleaves RNA and DNA. It doesn't really care, which is kind of fun. And what it basically does, you can think about it as like a Pac-Man. I don't know if I can draw a Pac-Man sideways. There we go. It's a Pac-Man. You have a chain that comes in and it will literally close and cut it. So when this thing is, oh my goodness, I can do it. I really can. When this is open, see the mouth is totally open. DNA can dock in and then the mouth closes, assembling the active site that's going to do the chemistry, cleaves the DNA, and then it opens back up. So the opening and closing motion is really important for getting substrate in, locking down the active site, and opening the active site back up. So opening and closing, those dynamics are really important for function. We've also seen other examples where dynamics are important. So we talked about this in an earlier video, the the glucose transporter. And in the glucose transporter, you can import glucose into cells by having a transporter that fluctuates between an open form that's open to the outside and an open to the inside form, allowing glucose and glucose alone to go through this bilayer hole. Only possible because the protein exists in two conformations, two states that it can fluctuate between. And finally, we haven't talked about this one yet, but we'll get to that eventually in a video, and that is ATP synthase, which is the enzyme in our mitochondria that is important for making ATP. And it actually couples flow of protons across a bilayer to rotation of the enzyme to building ATP. It's just a mind-blowingly cool mechanism. And not only is it a cool mechanism, but we can actually observe it in a test tube or in a microscope. What we're actually seeing here is a long chain of actins labeled spinning around in response to the generation of a proton gradient, basically. So we can actually see the motion under a microscope. So again, protein motion is really important for protein function. Last couple things I want to point out on this is that enzyme catalysis as a specific type of motion, so enzyme activities, we're going to talk about a lot of enzymes over these videos that catalyze particular reactions. They tend to go from 0.1 microseconds to 0.1 seconds, so they're in this regime. And if we bracket this, what we actually see, this is kind of on the slower end of protein motions, right? We've got the fastest motions over here up to the slowest motions. And also just as sort of useful sort of mileposts in your mind, we can think of catalase as one of the fastest enzymes over here, and Rubisco is one of the slowest, and you can see this is their sort of things that they peg out. But what's interesting is this is all way faster than biology still. So, right, the slowest enzyme is still quite a bit faster than, say, a cell dividing. And this is because this leads to and allows this separation of time scales, right? So we can talk about scales. We can talk about what's happening in proteins Lots of fast, interesting things are happening. But when we think about how that translates up into a cell, what we're actually thinking about is some kind of average of those behaviors. So we could think about it this way. So what I've done here is I've run a simulation of a protein, the staph nuclease again, and I've just overlaid a bunch of frames. That's kind of what's happening in a cell or what's happening in a test tube. Yet when we solve a structure, what we do is we average over all that and say, here's the structure. So whenever you're looking at a structure, what you're doing is you're actually looking at some kind of average of a bunch of different fluctuating structures. And in fact, we look at averages, you can actually see this sometimes explicitly. So this is a really fun example. This is using the technique of cryo-EM microscopy. And what the researchers have done is taken a bunch of cryo-EM images of circles, which are nucleosomes, these large protein particles that bind to DNA. And then these nucleosomes have DNA coming off it. So that's what these long chains are here. And what they wanted to do was figure out, well, what's the structure of the DNA in the nucleosome? How did they do it? Well, they've got thousands and thousands of pictures of different orientations of these molecules, which means they can do something called class averaging. So I'm going to zoom up on one of these here. So this right here, they said, we're just going to average every structure that clearly looks like it's from the top. And we're going to see interesting, see what pattern emerges when we average and average and average. They went over here to this one. This is one that was clearly from the side. This is one that clearly has the DNA wrapped and coming off. So they're averaging all of these pictures, using that to build up a three-dimensional version of what the nucleosome particle looks like. So what they've done is actually created something called electron density. This density here is basically where the average electron scattering is for the electron microscopy that says this is where the parts can go. So then the researchers can thread, in this case, they're threading nucleic acids through that blob, saying this is where the DNA went. And then in this region down here, they're threading in protein sequence to see where that protein went. And so they can learn something about how that nucleosome binds to DNA. But from the process as we've been thinking about it, what they're doing is they're just averaging over all the flopping, right? This is just an average over all of the flopping things that this molecule can do. They're taking something that's moving and they're building a static picture. And so I think it's just really important to keep in mind, this is a quote that I love from a paper from 1975, that a protein structure is a platonic protein, well removed in its perfection from the kicking and screaming stochastic molecule we infer must exist in solution. So that's what I keep in mind. Proteins, even though we're looking at these beautiful frozen structures, they're all moving, and as they're moving, that's really important for their function. All right, so let's quickly summarize. Proteins are always moving, and they're doing so on many time scales from like 10 to the minus 15 seconds all the way up to like 10 to the minus 3, so milliseconds. These protein motions are often important for function, doing things like closing active sites around things or letting molecules diffuse through a bilayer, for example. We can think about and visualize how these protein structures are how they're visited and sampled using a protein energy landscape. And finally, protein structures that we look at and we study are often static snapshots, but we always have to keep in mind that proteins are dynamic because that's how the proteins are actually doing whatever job they are that they're doing. So, all right, that's what I have on protein dynamics. This also concludes my block of videos looking at protein structure. I hope they were enjoyable and helpful, and I look forward to seeing you in future sets of videos.